Good morning everyone. Today I'll be presenting seminar on fibrin degradation products. I'll be dealing under the following headings. History. In 1937, McFarlane reported that damaged tissue release a substance activator that activate the inert precursor called plasminogen. Coming to introduction. Normal hemostasis. Mechanism by which bleeding from an injured vessel is arrested by formation of a thrombus. Its function is to maintain the blood in the fluid state, to prevent clots in intact vessels, and to arrest bleeding in the injured vessels. Components are blood vessels, platelets, plasma coagulation factors, and fibrinolytic system. Normal hemostasis. This is a flow chart showing normal hemostasis. Injury to the vessel wall leads to the constriction of blood vessels, which leads to the formation of temporary hemostatic plug. It also leads to the exposure of collagen, which causes platelet reaction and leads to the loose platelet aggregation, which in turn leads to formation of temporary hemostatic plug. It also leads to the release of tissue thromboplastin, which leads to the activation of coagulation and formation of thrombin, which in turn leads to the formation of definitive hemostatic plug. It takes place in 3 steps: tethering, activation and stable adhesion, spreading and aggregation. Coming to stages of hemostasis, it has 2 stages, primary stage and secondary stage. In the primary stage, there is injury to the vessel wall which causes platelet reaction and leads to the loose platelet aggregation and formation of platelet plug. In secondary stage, there is activation of plasma coagulation factors, which leads to the formation of stable fibrin clot. Then there is a dissolution of fibrin clot by fibrinolysis. Stages of hemostasis. In primary state, platelet and vessel wall is mediated. It occurs within seconds of injury. There is a formation of platelet plug and it prevents blood loss from capillary, arterioles and venules. Secondary stage, it is coagulation factor mediated. Takes several minutes for completion. It forms stable fibrin plug and prevents blood loss from large vessels. Fibrinolysis. Here the plasminogen is converted to plasmin by plasminogen activators, those that are kallikrein, factor 12A, tissue plasminogen activator, urokinase and streptokinase, which causes degradation of fibrin to fibrin degradation products and D-dimers, which has inhibitory effects on clot formation. The fibrinolytic system is activated in response to the activation of coagulation cascade. This system functions to remove fibrin from the vascular system in a controlled manner once it has fulfilled its function and to prevent excess fibrin accumulation. This system, which digests the fibrin clot through proteolysis, must be regulated so that proteolytic activity is limited to the area of fibrin formation and does not occur prior to the healing of the vascular lesion. Formation of fibrin. The insoluble fibrin clot is formed from soluble fibrinogen in 3 distinct steps. First step is the proteolytic cleavage of arginine and glycine bonds by thrombin, releasing fibrinopeptides A and B from the chains, forming fibrin monomer. Then there is spontaneous polymerization of fibrin monomers to form fibrin polymers. Then there is stabilization of fibrin polymers by factor 13A catalyzed cross-linking. Fibrin formation occurs both intravascularly and extravascularly as a consequence of coagulation, inflammation and tissue repair. When no longer needed, fibrin must be removed so that the normal vessel and tissue structure and function can be restored. The process of removing fibrin is called fibrinolysis. The fibrinolytic system is responsible for dissolving thrombi and maintaining a patent fibrinolytic system. The key components in this system are inactive proenzyme plasminogen activator, the enzyme plasminogen, fibrin and fibrin degradation products, inhibitors of plasminogen activators, and plasmin. Fibrinogen. The fibrinogen molecule is a large trinodular glycoprotein found in plasma and platelet alpha G. Megakaryocytes and platelets do not synthesize the protein but absorb it from plasma. Fibrinogen is the most abundant coagulation protein with a plasma concentration of 2 to 4 microgram per ml, representing about 2% of the total plasma protein concentration. Fibrinogen is encoded by 3 separate genes located on chromosome 4, and the production of the 3 genes is coordinately regulated. Fibrinogen is also one of the several hepatic proteins whose production is increased as a part of acute phase response mediated by interleukin 6. This is the structure of fibrinogen. Fibrinogen is a trinodular structure composed of 3 pairs A alpha, B beta, gamma of disulfide bonded polypeptide chains. The central nodule is known as E domain. Thrombin cleaves small peptides A and B from the A alpha and B beta chain in this region to form fibrin. The central nodule is joined by supercoiled alpha helices to the terminal nodules, also known as D domains. Release of FPs A and B. Thrombin binds to the central E domain of the fibrinogen and cleaves specific arginine glycine bonds, releasing 4 peptides from the fibrinogen molecule. It releases a short peptide containing 16 amino acids from each A chain, that is fibrinopeptide A, FPA, as well as 14 amino acid peptide from each B chain, fibrinopeptide B. The resulting molecule is called fibrin monomer. Release of FPA alone is sufficient for fibrin assembly process. Assembly of fibrin polymers. The cleavage of FPA and FPB exposes binding sites in the central E domain that interact with complementary sites on the gamma chain of the D domain of the other fibrin monomers, creating a DE contact. The polymerization sites on the D domain are always available and interact with complementary sites on the E domain after thrombin cleavage. The polymerization continues by addition of a third monomer forming a DD contact as well as another DE contact. The use of X-ray crystallography to study fibrinogen has provided insights in the mechanism by which fibrin self-assembles. The crystal structures show a mechanism for electrostatic steering to guide the alignment of the fibrin polymers during polymerization. The non-covalent interaction of the nodules leads to the initial formation of 2-stranded polymers termed protofibrils. The polymer strands aggregate in an overlapping pattern called half-staggered array, in which second strand is offset from the first by half of the length of the single fibrin molecule. The protofibrils then aggregate into thick fibers through lateral association held together by weak non-covalent and electrostatic interactions. During fibrin monomer polymerization, other plasma protein including components of fibrinolytic system, that is plasminogen activators and fibrinolytic inhibitors, can also bind to the fibrin surface. Thrombin extension in vivo can be blunted by unactivated fibrinogen molecules or fibrin degradation products, which can bind to and cap the ends of protofibril. Now coming to fibrin stabilization. Spontaneously polymerized fibrin strands are unstable, but the alpha and the gamma chains of the adjacent fibrin strands are subsequently covalently cross-linked by F factor 13A. The cross-linking strengthens the cloth and provides the resistance to the chemical 5-M urea and enzymatic plasmin digested compared to the uncross-linked polymer. Now coming to the formation of fibrin polymer. Thrombin cleaves the A and B fibrinopeptides from E domain of the fibrinogen to form fibrin monomer. The cleavage apparently changes the negative charge of the E domain of the positive charge. This permits the spontaneous growth of fibrin polymers as the positively charged E domain assembles with the negatively charged D domains of other fibrin monomers. The polymer is initially joined by hydrogen bonds. Factor 13A in the presence of calcium ions is responsible for catalyzing the formation of covalent bonds between glutamine and lysine residues of adjacent monomers, that is D domains, thus stabilizing the lattice formation. Factor 13. The final reaction in fibrin formation is the stabilization of fibrin polymer catalyzed by factor 13A. Factor 13 circulates as heterotetramer alpha 2 beta 2, which is the product of two separate genes coding for the A and B chains. Factor 13 is found evenly distributed between plasma and platelets. Platelet factor 13 exists as an alpha 2 dimer lacking the B subunit. The plasma factor 13A chain is primarily synthesized by bone marrow cells, megakaryocytes, monocytes, macrophages, and is stored in the soluble non-granular fraction of the platelet. The A chain contains the active site of the enzyme, the fibrin binding and substrate recognition domains. The B chains are secreted by hepatocytes and complex rapidly with the A chain in the plasma, stabilizing them and prolonging their plasma survival. The B chain also promotes plasma factor 9 association with fibrinogen. Plasminogen and plasmin. Plasminogen is a single chain glycoprotein synthesized by the liver. It has 5 Kringle domains that are critical in regulating fibrinolysis. The Kringles contain lysine binding sites that are responsible for binding affinity of plasminogen and plasmin for fibrin, cell surface receptors and other proteins including alpha-2 antitrypsin. Plasminogen preferentially binds to C-terminal lysins as opposed to intramolecular lysins. Plasminogen activators convert plasminogen into active 2-chain form, the serine protease plasmin, by hydrolysis of a single peptide bond. The active enzyme plasminogen has a positive feedback effect on fibrinolysis. When plasminogen is activated, it unfolds to expose its potential enzymatic domains. Here in this figure we could see the closed form and the open form. Coming to activators of fibrinolysis, the two major plasminogen activators that occurs in the circulating blood are tissue type plasminogen activator TPA and the urokinase type plasminogen activator UPA, also known as urokinase. The contact phase of the coagulation produces serine proteases, that is factor 12A and kallikrein, which also can promote plasminogen activation. The fibrinolytic system is the following. The fibrinolytic system can be activated by physiological activators, tissue plasminogen activator derived from endothelial cells and the urokinase type plasminogen activator, or the contact activators kallikrein, factor 12A and factor 11A. Exogenous activator streptokinase can also activate the system. Plasmin, the product of activation, controls coagulation by digesting fibrin to fibrin degradation products and by degrading factor 5, 8 and factor 12. The inhibitors of plasminogen activation are plasminogen activator inhibitor 1, plasminogen activator inhibitor 2, plasminogen activator inhibitor 3, thrombin activable fibrinolysis inhibitor TAFI, and inhibitor of plasmin, and serve to control the fibrinolytic system. The fibrinolytic system, like the clotting system, is intricately regulated by these activators and inhibitors. Now the fibrinolytic system, it cleaves the fibrin and now we are dealing with the role of plasmin. The plasmin will cleave the fibrin and fibrinogen to fibrin degradation product X, Y, and DE. It activates fibrinolysis, and it activates intrinsic coagulation pathway. It converts factor 12 to 12A is amplified indirectly by plasmin. It interferes with intrinsic and common pathways, that is, it destroys factor 8 and factor 5. It blocks the thrombin conversion of fibrinogen to fibrin, that is, fibrin degradation products interfere with thrombin influence on fibrinogen. Here there are, in this diagram we could see fibrinolytic mechanism. In the figure 1 we could see the damaged endothelia secrete tissue plasminogen activator. The tissue plasminogen activator activates plasminogen bound to fibrin clot. Then the free tissue plasminogen activator circulates bound to PA1 and is cleared from the plasma. Here in the second figure we could see plasmin systematically hydrolyzes lysine and arginine peptide bonds of the fibrin polymer. Here the P indicates sites where plasma cleaves the fibrin polymer. Coming to fibrin degradation, plasminogen is responsible for asymmetric progressive degradation of fibrin or fibrinogen, forming distinct protein fragments referred to as fibrin split degradation products or fibrin split products. The liver rapidly clears these fragments from the circulation. Their detection in the plasma is of diagnostic value for some hemostatic disorders. This is the plasma degradation of fibrinogen. The degradation of fibrinogen by plasmin occurs in well-defined sequential steps. First, small peptides are cleaved by the carboxyl ends of the alpha chain, producing fragment X. The E domain retains the A and B peptides. The fragment X is still capable of reacting with the thrombin to form fibrin. Next one of the D domain is cleaved from the fragment X, producing fragment Y, that is DE, and fragment D. Further cleavage of the fragment Y produces D and E fragments. Now what is D-dimer? D-dimer is a protein that represents an area of fibrin which contain cross-linked regions used as clot stabilizers. D-dimer present in circulation is used as an indicator of a blood clot being formed and broken down somewhere in the body. A test for breakdown products, the D-dimer test, is a specific marker for plasminogen degradation of fibrin. The fibrin fragments, if present in sufficient concentration, can exert an anticoagulant effect on the clotting system. Fragment X can still react with and bind thrombin, but very slowly. Fragment X can compete with fibrinogen for thrombin so that less fibrin is formed. Fragments Y, D, and E inhibit the polymerization of fibrin monomers. The fragments can also interfere with primary hemostasis by inhibiting platelet aggregation. Here we could see the schematic drawing which depicts some of the derivative of plasmin digestion of fibrin. Fibrin digestion by plasmin occurs at a cleavage sites identical to those of the fibrinogen. However, because of the covalent bonds of the factor 13A stabilized polymer, digestion is slower. Derivatives also are different at some sites are not accessible in the lattice formation. Now coming to pathological effects of fibrin degradation products. Fibrin degradation products are significant because of their hemostatic effects, which include antithrombin activity, interference with polymerization of fibrin monomer, interference with fibrin activity. The early and the large fragments X and Y, along with intermediate fibrin degradation products, are important in exerting anticoagulation effect. The fragment Y and D inhibits fibrin polymerization. Fragment E is a powerful inhibitor of thrombin. All four fragments, but particularly low molecular weight fibrin degradation products, have affinity coating platelet membrane and therefore cause a clinically significant platelet dysfunction by inhibiting aggregation. Now coming to fibrinolytic inhibitors. First one is alpha-2 antitrypsin or alpha-2 antiplasmin. Alpha-2 antiplasmin. It is an alpha-2 glycoprotein, most important naturally occurring inhibitor. The principal inhibitor of fibrinolysis is by binding with plasmin that is free in the plasma, neutralizing plasmin. It inhibits the clot promoting activities of plasma kallikrein. It inhibits the serine protease 12A, 11A, 2A and 10A. Hereditary deficiency have been associated with excessive clotting, that is disseminated intravascular coagulation, and excessive fibrinolysis. Second is alpha-2 macroglobulin. It is large naturally occurring plasma glycoprotein. It inhibits component in both the fibrinolysis and coagulation systems. It inhibits plasmin after alpha-2 antiplasmin depletion. Third is alpha-1 antitrypsin. It is the third most important naturally occurring inhibitor of fibrinolytic system. It activates plasmin slowly and does not bind plasmin until both alpha-2 antiplasmin and alpha-2 macroglobulin are saturated. It inhibits coagulation by its potent inhibitory effects on factor 11A. The other inhibitors are antithrombin 3, which inhibits fibrinolysis by inhibiting plasmin and kallikrein. The C1 inactivators also inhibit plasmin. Now coming to downregulation of plasmin formation by thrombin activable fibrinolysis inhibitor. Downregulation of the fibrinolysis is initiated when plasminogen is converted to plasmin by tissue plasminogen activator. Both plasmin and tissue plasminogen activator preferentially bind to carboxy terminal lysine K residues of fibrin. The formation of plasmin is enhanced by a positive feedback loop. New C-terminal lysine residues generated after the limited plasmin cleavage provide two binding sites for continued tissue plasminogen activator and plasminogen binding. The thrombin activable fibrinolysis inhibitor, that is TAFI, cleaves off the C-terminal lysine residues from partially degraded fibrin and thereby eliminates or decreases the fibrin cofactor function in tissue plasminogen activator or plasminogen or fibrin complex, inhibiting the formation of plasmin and the degradation of fibrin clot. Now coming to fibrin degradation products assays. Here the plasminogen is converted to plasmin, which in turn leads to the conversion of fibrinogen or fibrin to fibrin degradation products. The principle of it is a suspension of latex particles sensitized with specific antibodies to fibrin degradation products fragments D and E. Suspension mixed on a glass slide with dilution of test serum. Agglutination indicates presence of fibrin degradation products. Interpretation of FDP assessment. Agglutination with 1 in 5 dilution, that is FDP more than 10 microgram per ml. Agglutination with 1 in 20 dilution, that is FDP more than 40 microgram per ml. Normal range is less than 10 microgram per ml. 10 to 40 microgram per ml indicates acute myocardial infarction, acute venous thromboembolism and acute pneumonia. And if it is more than 40 microgram per ml, then it is disseminated intravascular coagulation, thrombolytic therapy with streptokinase. Now, utility of FTP assays. The following conditions are associated with increased FTPs, that is liver disease, alcoholic cirrhosis, kidney disease, cardiac disease, post-surgical complication, carcinomas, myocardial infarction, pulmonary embolism, DVT, eclampsia and disseminated intravascular coagulation. Coming to recent updates on FTP assays, abnormal coagulation parameters associated with COVID-19. The medical team observed the coagulation test results and disease outcomes in subjects with confirmed COVID-19. The patient having markedly elevated D-dimer and FTP results went hand in hand with the higher risk of mortality associated with COVID-19. Elevated levels of D-dimer and fibrinogen are common markers of the disease state and are proportional to increased levels of CRP. Elevated PT and APT times are also observed. Level of fibrinogen less than 100 mg per dl can be seen along with elevated D-dimer levels. The elderly and the subjects with comorbidities have higher D-dimer levels and have a greater chance of dying from COVID-19 virus. Now coming to D-dimer assay. The D-dimer is a specific marker resulting from plasmin degradation, that is lysis of fibrin clot, which has been cross-linked by factor 13A. D-dimer is an excellent marker for disseminated intravascular coagulation with secondary fibrinolysis. The D-dimer test utilizes monoclonal antibodies against the D-dimer fragment. It is a semi-quantitative assay that utilizes macroscopic latex agglutination. The assay can be performed on plasma, citrated EDTA or heparinized and serum. The extra step of preparing serum as a standard FTP test is avoided because fibrinogen and fibrin do not cross-react with the D-dimer monoclonal antibodies used in these tests. The patient specimen is mixed with latex particles coated with monoclonal antibodies directed against D-dimer on a glass slide for a specific amount of time, that is 2 minutes. At the end of this time, the reaction mixture is observed macroscopically for the presence of agglutination. Most procedures test both undiluted plasma and a plasma sample diluted 1:2 in buffer, which allows a semi-quantitation of results. Lack of macroscopic agglutination in either sample correspond to a D-dimer level of less than 0.5 microgram per ml. Macroscopic agglutination in undiluted sample but not in 1:2 dilution corresponds to a D-dimer of 0.5 to 1 microgram per ml. If both samples shows agglutination, the result is more than 1 microgram per ml. Normal individuals have less than 0.5 microgram per ml of D-dimer. Causes of elevated D-dimers: disseminated intravascular coagulation, trauma, surgery, hematoma, arterial thrombosis, pregnancy, cancer, diabetes, thrombolytic therapy, and older age. Now coming to summary of properties of the components of the fibrinolytic system. The components are under the following headings, that is fibrinolytic component, activators of plasminogen, inhibitors, and the cell surface receptors. In fibrinolytic components, there are plasminogen and plasmin. Plasminogen is a single-chain glycoprotein and it is a precursor of plasmin. Plasmin is a dimeric glycoprotein which cleaves fibrin. The next is the activators of plasminogen, that are tissue plasminogen activator and urokinase type plasminogen activator. Tissue plasminogen activator is a glycoprotein which is complex with inhibitor plasminogen activator inhibitor 1 which activate plasminogen. And the second is urokinase type plasminogen activator, which is a single chain glycoprotein. It is low enzymatic activity until cleaved to tissue plasminogen activator by plasminogen, and it activates plasminogen. And the inhibitors. Inhibitors are plasminogen activator inhibitor 1, plasminogen activator inhibitor 2, alpha 2 antitrypsin, thrombin activated fibrinolytic inhibitor. Plasminogen activator inhibitor 1 is a single chain glycoprotein. It is protease inhibitor, that is serine protease inhibitor serpin. It inhibits tissue plasminogen activator and urinary plasminogen activator. Then the plasminogen activator inhibitor 2. It is a glycoprotein and it is a serpin, that is serine protease inhibitor, which inhibits tissue plasminogen activator and urinary plasminogen activator. Then alpha 2 antitrypsin, which is a single chain glycoprotein. It complexes with plasminogen and also inhibits kallikrein, thrombin and tissue plasminogen activator. And the next is thrombin activated fibrinolysis inhibitor. It is a single chain protein and it inhibits plasminogen activation by cleaving lysine from fibrin. And the fourth category is cell surface receptor, that is UPAR, that is urinary plasminogen activator receptor. It is a glycoprotein receptor. It binds to urine plasminogen activator, localizes on surface of endothelial cells, monocytes and macrophages. Then annexin 2, which is heterotetramer, it binds to tissue plasminogen activator, plasminogen, and it is found on endothelial cells, monocytes, macrophages and smooth muscle cells. And the next is low density lipoprotein receptor like protein LRP. Its role is clearance receptor for urine plasminogen activator and PAI1 and tissue plasminogen activator and plasminogen. Here are my references. Thank you.